and away we go. Go ahead.
All right. Okay. Thanks. I'm going to put my screen up for everybody to share, everybody to see. All right. So what we're going to talk about today is compartmental modeling, and this is the last of what I kind of would refer to as the boring kind of let's get all the kind of learning all the players, learning the words, learning the phrases and the players of pharmacokinetics out of the way. And what I'll start with the next lecture is probably I want to talk about physiological differences in kind of adult and pediatric patients, and mix in some of the physiological differences associated with veterinary patients as well, both to consider how animals differ for humans, both in the drug development kind of pipeline, as well as the fact that you know, there a lot of us are interested in kind of one cure type things, and how we can use these same drugs to treat some of our veterinary patients. So not only just from the kind of development perspective, but also from the animals as patients perspective. So that's what we're going to do in April, and it's going to be a lot more exciting for all of us. But we do need to, I mean, I think it is important that I go through this stuff, so when I talk about data and studies and stuff like that, everybody knows what this data comes from. Okay. So we talked about non-compartmental modeling in the last lecture, and what we're going to talk about today is compartmental modeling. And the difference is really in non-compartmental modeling, you think of every data point as its own entity, whereas in compartmental modeling, we collapse all the data into a curve, and the curve and the equation that represents the curve is really the data point, the data set. So you coalesce kind of all of that, the time course data into a curve that's represented by an equation. And that's illustrated here in NCA, where we'd use trapezoids to figure out the area under the curve, and you'd take each point and kind of do the math down to calculate AUC. Here's that same data, but now I've actually done a nonlinear regression and fit the best line here. You get rid of those points, and there's your data. So that representative equation actually is going to allow you to calculate all your points. So it's a way really to smooth out the points, to recognize the connectivity between early and later time points, and the fact that it is a continuum from dosing and elimination. So it allows this kind of cleanup step and the data to kind of be kind of better represented as a continuum, which it really is when you give a dose and you're looking at the time course of drug loss and metabolism and distribution in the body. So it's important to remember that when we do these parameter calculations, they're calculated based on the data describing the curve fit, and that the data points kind of become the curve. And so for keys to compartmental modeling, they're data-based, so it's all needs to be, you know, the curve fits the data. It relies on curve fitting and assessment of goodness of fit of that curve. That means we can't do it in Excel like we did in the non-compartmental modeling in the last lecture last month. You need to use some type of software program, and the kind of industry standard used to be called WinNonLin. Now it's Phoenix WinNonLin, and this is a nonlinear curve fitting package that also has all the equations and calculates, you know, all your goodness of fits and Akaike information criteria and all the type of things that you need. These curve fittings can also be done on things like MATLAB. It has a kind of a biology component, a biomodeling module that includes some PK modeling. There are various R-based and other like ADAPT type programs. ADAPT comes from the University of Washington, that you can also do this type of curve fitting. So there's a lot of ways to do curve fitting, but the point is it really requires a specialized nonlinear curve fitting software. Okay. The other thing to remember about compartmental modeling is that it requires both an input for your dosing and an output, your distribution, equilibration, and elimination. And I'm going to talk a little bit about that because it's important to note that you also need to have the end component. You just can't dump it in there or not take it into account. In non-compartmental modeling, you really can. You don't really need to worry about modeling the in part because you just take on the up part, you handle the points the same as you do on the down part. Whereas in compartmental modeling, you have to come up with an equation that describes the drug delivery as well as the elimination. And so luckily these things have been looked at for a long time, and they're relatively standard. People have looked at about every situation you can possibly imagine, and it's all kind of there. And in a lot of these software packages like Phoenix non-win non-len, you can just go pick the one you want. IV infusion, extravascular dosing, whichever one you want, without a lag time. MATLAB also does that, where you can kind of have this plug and play with regards to the equations associated with the dosing. Okay. So let's start with some data. So here's some data that you may get. So you do a PK study, you get some plasma, you send it to the lab, we measure it, and we send you the data back. And you graph it, and it looks like this. And that's always where you start. You need to graph it. You look at that and you go, okay, what do I do with that? Is this compartmental or non-compartmental? And the thing you have to do is you have to try to fit it. Okay. And usually you'd start simplest and work up. And the simplest compartmental model is a one compartmental model, which basically treats the body as one well-mixed system. Now, that doesn't mean that the drug is at the same concentration everywhere in the body. It just means that functionally it looks like that. So anytime you take drug in the blood and other tissues, instantaneously equilibrates, and so you really can't see any difference in those. And so it functionally acts as a single component, or what we call a one compartmental model. So you have a dose, which is the input. You have a volume of that one compartmental model, which for one, or for that compartment, for one compartment, it can be called the volume of one, or the volume of distribution is the one you'll hear most commonly. And there's also elimination. So you got the in and you got the out. Okay. Now we're going to start with a bolus IV dose. So all the dose goes in at time 0, and that's by far the easiest. You just dump it in at time 0. Okay. So bolus IV is where we're going to start. The concentration in the blood is actually symbolized or calculated as the amount in the body divided by the volume of distribution. And so the concentration at any time 0 is going to be the dose divided by the volume of distribution, because at time 0, none's been eliminated, all the drug is there. So you can actually calculate a concentration at time 0 if you know the volume of distribution for a given drug. And that becomes really, really handy, because you can actually use a dose, if you know this, to target exactly a plasma concentration that you want. So this really allows you to start doing some very defined dosing and targeting specific concentrations when you have some of these parameters, once you have PK data. And that's kind of using the PK, not defining the PK, but I think it's important to point that out, is that's why that's important, and that's why we care. Because one thing you do want to know is, if I give this dose, what concentration am I going to get? And the volume of distribution is the parameter that allows you to go from dose to concentration in the blood or plasma. Okay. So one thing, and I do this because, not to, I don't want people to know the math, but I have to go through it. But I do think it's important to know this is based in first principles. Okay. And so when you're talking about a one compartmental modeling and you're talking about drug in the blood, it's in the blood, it gets metabolized or eliminated as this little red dot, symbolized leaving. And that represents excreted drug, and that's why the curve goes down, as you're losing it from the body. Now, when you're measuring, what we're actually looking at is loss of drug in the blood, and that loss is actually equal to drug in the blood times KEL. Because the other thing that's inherent to these models, these are linear models, is that you have first order elimination, or that elimination is directly proportional to the amount of drug that's there. Okay. And so these first order processes kind of are a general standard in drug metabolism. They're the way things are for the most part for 90% of drugs. When you start seeing saturation and you don't have first order processes, that's when things can get very complicated. But luckily, for the most part, most drugs are eliminated by these first order processes that are um, uh, the more drug you have, the more elimination you have. It's proportional to the amount of drug. So it's just drug in the blood times this elimination rate. So mathematically, if you look at this, the change or loss in drug in the blood is equal to the concentration in the blood times the KEL. If you do a little rearranging here and you wind up with DC blood over concentration blood, if you bring everything over to the same side, equals negative KEL times DT. If you integrate this, you integrate both sides, you integrate from DC to C and DT. When you pull this out, you wind up, when you do that math, the integral of DC over C, changing anything over itself, is actually the log of that change over the original value times negative KEL times T. If you just rearrange and solve for the concentration and E times T, it's CT equals C naught times E to the negative KT. So the reason this is important, okay, is that this explains all first order processes, okay? And this is the exact same equation that you would use for looking at the decrease of a radionuclide over time, with its half-life being this KEL, or being related to this KEL. You could figure out exactly how much radioactivity you have at any time if you know how much you start with and you know the half-life. That's effectively the same system we're working at. When these first order processes do that, you wind up with this exponential relationship between time and concentration associated with some elimination process, which is related to a half-life. Okay. Now, I kind of mentioned it last time. Well, we'll see it in a minute when we calculate the half-life. But the point is that this concentration any time T equals C naught times E to the negative KEL times T is something you're going to see again and again in the first order, in the second order, and the third order. They're always going to be the same. We're just going to add more of these phases. Okay. And so these model systems are really based on this kind of exponential decay model and first order elimination. Okay. So what this tells us, all right, is that if we log transform this curve that we got right here, you get a straight line. Okay, so if you click just on Prism, you have it linear, and you click on, let's make it log, you could either be log 2 or log 10, you're going to get a straight line. Okay, the reason is that this concentration E times T is logarithmically by E here related to KEL times T. And so if you plot CT versus T, you wind up with a straight line with the slope that's the KEL. Okay. And so the data fits a straight line with a slope of KEL and a y-intercept of C0. Okay, y equals mx plus b. Same basic thing you learned in basic geometry years and years ago for the equation for a straight line. And that's really what we're talking about here with the one compartment model. When you log transform it, you wind up with a straight model or a straight line because of that log relationship. Okay. Enough with the math. Why do we care about that? Well, because if we fit this, and if you fit this line, and I did it, I think I just did it in Prism, you get a y-intercept of 385 nanograms per mil, a slope of 0.0565 per hour. We'll go back and look at these in a minute. You can calculate the volume of distribution. It's dose divided by C0. Your volume of distribution in this case is going to be equal to 260 liters. Okay. The half-life 0.693 divided by KEL is about 12.3 hours. If we go back and look at this and say, does that make sense to us? Where's our data? Oh, I don't have, oh, we'll have to look here. About 12 hours. Well, 12 hours, we're here at about 200, right? And our C0 was at about 400. So does 12 hours make sense? Absolutely. All right. And if we go to 12 our 12-hour to 24-hour. 24 looks like about 100. This is 200. So every 12 hours, it drops by half. So the half-life being calculated at 12 hours makes sense relative to your data. It's always a good idea when you calculate anything to go back and make sure it makes sense with your data. If it came out to be 165 hours, you'd probably go, what? You'd go back and look at the data. Just don't go, well, the math said that. Figure out that you'd actually have to want to go back and look at it and say, okay, does that make sense with the data? Because this is purely should be a reflection of the data. Okay, half-life 0.693 divided by KEL, 12 and a half hours. Clearance in the one compartment model is volume of distribution times KEL. We calculated this here. We have our KEL up here. Calculate that at 14.7 liters per hour. The area under the curve, now we don't have to use trapezoid. It's actually C0 divided by KEL. That comes out to be 6,814 nanograms per mil times hour. The area under the moment curve, which is the second derivative with time. If you remember when we did it in the non-compartmental, we multiplied it by time. We just do that again. Now our KEL is actually just squared in the denominator. You do that, comes out to this value. And the MRT, just like in the non-compartmental, the mean residence time is AUMC divided by AUC, comes out to be about 17.7 hours. So the point is that once you fit these, once we got these values here, we're able to get this one, and work our way all the way through everything else. So the values of the curve fitting parameters, the C0 and the KEL opened up all the other parameters for calculation. So in some regards, once you get it fit, it's a lot easier than the non-compartmental to calculate things out. And it is, because you're only now working about with the equations that represent the curve, and you don't have to take every point independently when you do all these calculations. So you can figure out your PK parameters relatively quickly once you know what your equation fit is. Okay. Okay, so that's the one compartment model. The key here is that it fits a straight line when you plot it in log linear. Okay. And the interesting thing, and there's an example in Rowland and Tozer, which is kind of the pharmacokinetics textbook, like Goodman and Gilman is the pharmacology textbook. They have an example with some opioids. And actually, if you start at an hour, you get a straight line. If you get all these earlier time points, you get these other phases that really starts to show up. So the fit of this curve, if you go out long enough, you're always probably going to get just a linear straight line. It all depends on how much data you have in terms of getting these earlier phases. Okay. And that gets to multi-compartment models. Okay. So what if your data looks like this? Okay. So we put it on the log linear curve. Here we have a log scale on the y-axis, a linear time scale on the x-axis. You can see here that if I try to draw a straight line, I miss a lot of these points. I mean, because this one, it comes up and you go, well, that's not too bad a fit, except it hits C0 right around 400. And our C0 actually, at 5 minutes, it's over 1,000. So clearly we're not fitting this very well. And if I fit these 3 points here, I miss all these later points. So how do you draw a line there? You just draw a straight line that kind of cuts across the middle? Well, you could, and that would be an approximation, but you can be better. We can all be better. And the reality is that this is biphasic. It has one that comes down here and then one that comes here. And if you fit 2 lines to it, you can see pretty clearly, you can draw 2 relatively straight lines through here. If you add these lines together, and I got clever with my colors because I have blue here and red here, and blue plus red equals purple. If you add those, you get a line that fits all of this data quite nicely and describes it. And this is the basis of multi-compartment models. It's just adding these together. So now we have an early distribution phase where the drug's distributing from the blood into the tissues, and then an elimination phase where you're eliminating the drug from the body. And so that's what we call these, the distribution phase and the elimination phase. And it's really now 2 mono exponential things that are put together. So this is a 1 compartment model. If you get rid of the red here, this is CT equals C naught times e to the negative KT. All right. And we'll call these, since they're the first part, A and alpha. And then you have the second curve, which is now beta. So it's still also some C times e to the negative KT, except you have a different starting point and a different KEL. And when you add those together, you get this very nice fit of this. So a two-compartment model is a multi-compartment model is exactly that. It's just fitting these different phases with curves that are independent. And when you add them together, they describe the data. Okay. Now in practice, you could draw these straight lines. It gets really difficult, and that's how curve stripping used to be done. Now, again, we have software packages that will strip the curve quite well and compare them for you and tell you which one to use. If you go to a three-compartment model, and here's some data now that fit a three-compartment model where we can actually draw three lines here. Now you have an equilibration phase to go with the distribution phase, and now you're just going to be adding another mono exponential equation that's going to be C and gamma. Okay, so here's that data here. Fits a nice three-compartment model. Okay, and when you put that together, you get this. Here's a green line that's just all of those lines put together, and you can also describe this kind of tri-exponential or three-compartment. Okay, this is actually a representative diagram. This you'll see when in one of my examples, when the math is actually there, there's these transfers. So unlike a one-compartment model, the three-compartment model, you actually don't have the same rate of transfer or instantaneous movement from other parts of the body. There's some that are slow. So you could think of one as a shallower or a deeper compartment, whether it be fat, where the drug kind of sticks and comes out more slowly because of low blood flow or something else. But you have these compartments where drug doesn't instantaneously back equilibrate with the blood, but kind of is a little slower coming out, leading to this slower elimination phase. So the movement between compartments is responsible for these different phases. Elimination is again presumed to occur only from the central compartment. The compartments do not actually represent any actual tissue compartment, but rather areas where drug moves away from the central compartments, which includes the blood. So I think, and this is important because people think about talking about these as actual organ systems. They're not. There is a specific type of pharmacokinetic modeling where you do that. That's called physiologically based modeling. We will talk about that because that's actually a quite advanced way to look at some drugs and their metabolism, and you can do some pretty cool stuff with it. So they're just kind of ways to make this shape mathematically. So again, it's all data based. Data looks like this. We got to find a way to do that. This is the way to do that. Okay. Luckily, three compartments is generally the maximal number of compartments. That's partially because any more complexity, you probably want to get into a physiologically based model. The other thing is that to fit different phases, you have to have a lot of different time points. I mean, for these three phases, I got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 time points. If you start taking that many time points, you're probably not going to have many compliant patients. And so, to fit many more points, it's just really not possible. So the most you're going to see, and when I get to examples, the most you're really going to see are kind of three compartment fits. Okay, so that's kind of the maximum. Okay. So the point of the first part of this lecture is just kind of talk about and emphasize that these compartments are data-based and a way to describe the data that we get. We go from the very simple one compartment model, which is a straight line, which involves really this kind of single system, single compartment that's instantaneously mixed, and the body acts like that, such that you really don't get any type of intercompartmental transfer that leads to distribution and equilibration phases. More complex models are two compartment, and now we have to add a distribution phase, which is represented by a curve up top, and then an elimination phase. When you add those, you get this nice fit, and it's just now adding another exponential equation. So you have two of these mono exponentials going together. And if you get more complex three phases, then you just have to add another one, and you have three phases or three mono exponentials, and that's about the extent of what we do for compartmental modeling. Okay. So why do we do this? Why do we do it this way? Well, once we start having equations, we can start mucking around with things. And so when you think about this, if you have an equation where this represents distribution, this equilibration, and this elimination, let's say you say, what would happen if you had an individual that had two drugs on board and you had competition for metabolism? You'd probably think that it was going to be in this elimination phase, and you could play around and see what happens if you change this very much. So you can actually start kind of mimicking what would happen if you had drug interactions. This is more adaptable because you have kind of plug and play things that you can start playing with once you have these equations that describe the PK. And I'll kind of talk about some of these, as some of this is done and looked at in the examples. So what I'm going to do for the rest of the lecture is really go over examples of how this stuff is used, which I think will really segue into the next lecture about physiological differences and why you can actually attribute some of the things that are measured to physiological differences and or changes in the individuals that we're talking about. Okay. So what about drug dosing? I talked originally about IV bolus, but the cool thing is that we can actually add. This is the exact same model, but instead of adding it all at once, now I'm adding a dose that has an absorption component. So this would be oral dosing. In this case, this is a drug called ABT-116 that was being developed for pain as a veterinary drug. And we did a study here a number of years ago now in collaboration with our anesthesia group. And this is the actual data from 6 dogs. When I fit a one compartment model with first order oral absorption. And what you can see here is that this is really right here your elimination. F times dose is C0 for an oral. F is bioavailability, and so your bioavailability times your dose is going to tell you how much gets in divided by volume of distribution. So that's kind of your C0 component. But C0 is going to be affected because you're going to have absorption elimination going at the first time. So you have to adjust that, because as you have drug being absorbed, you're going to be eliminating it. So you're not going to get all a C0 that's complete because of the fact that when you absorb it, all some's already going to be eliminated. So you have to account for that mathematically, and that's what that does. And then you have absorption going on and elimination at the same time. When you put this together and you solve this equation, you get this curve here, which pretty nicely represents the data. You miss these points, but if we had all the points in here, you'd see it hits the median ones here. It's just the way it looks. And you can see we get an area under the curve. We can calculate a Ka, so we can actually get a half-life for absorption. Why is that important? Because you can estimate how fast you're going to hit your max or how fast the drug's going to get on board, which may be important if you need a drug to act fast, which for a pain med would be recommended. You don't want to have to wait 3 days for it to accumulate to get any control of pain. In this case, there's a lag time associated with this, which is accounted for here. And we know that it's about, half an hour. That's not all that good for a pain med, but not too bad, as it does start to come up relatively quickly. You know how long it reaches max? That's good. It reaches max in about 4 hours. That's when you should have your maximal effect, you would think. So you can actually look to see if your effect matches your drug concentration or if it's working in a central compartment. If it works in the brain, you can see that. With opioids, usually you see a delay in that. So you can actually start then thinking about how the drug's working. This data can start helping you think about the drug, what activity you're getting and how that activity works. Your half-life, which is critical to start thinking about redosing. Half-life 6 hours, so at 12 hours, if you're dosing BID, it's going to be down, a fair amount. And actually, you can use this data and start simulating other doses. And that's actually what we use this data for, to figure out if we wanted to keep the drug above a certain concentration that we thought it needed for pain threshold. What dose could we give and how often should we give it? The reason we wanted to do that is to actually go forward with a trial to test this as a pain med in dogs. And so we worked out those doses, and then they were able to go forward with those. I think it was 20 mg per kg BID worked quite good, even though the single dose for the PK was done at 30 mg per kg. We could simulate using these parameters and guess what it would happen, what it would look like, pretty simply by just using the same equations but plugging in a different dose, just changing this dose and simulating what that looked like. So this compartmental structure gives you that ability to simulate these pretty simple systems, to work out different doses, different dosing intervals, and all kinds of different things. Okay, so the cool thing is you can add these different kind of inputs. Okay, here's another example. This is a pharmacokinetic study with paclitaxel this was published in 2003. Um, and the key here in the modeling is that this is one of the first times we actually measured paclitaxel, or I'm sorry, this is not this is docetaxel. I, I'm even the first. I even wrote this paper and I can't even remember that. This is docetaxel, not paclitaxel. But the cool thing about this was that this is one of the first times we used LCMS or mass spectrometry to measure docetaxel, and we were able to measure it out to much later time points. And the point here that we try to make in this paper is that if you predict the half-life only going out to 24 hours, which is about all you could do with typical HPLC methods, the half-life you predicted was 15 hours. If you look out at 24 hours, or if you can go out with a 48-hour time point, you can actually measure it more accurately to be about a day. It's about 24 hours. So, and that makes a difference. And that difference is shown when you predict the concentration of the drug at later time points. You would see that it goes much lower. Why is this important? Well, the first lecture we started talking about thinking about chemotherapies as antibiotics, right? And you may have a minimum inhibitory concentration. And at the time, we were really interested in taxanes as potential in using them in kind of low dose, metronomic protocols. And do we need to keep them above a certain concentration to get inhibition of endothelial cell proliferation? And that's really what we were looking at. And these studies were in collaboration with Dr. Eckhart when she was here. And so we were really interested in more accurate predictions at later time points to keep docetaxel levels above what we thought was the concentration required to inhibit endothelial cell proliferation, which we thought was around a nanomolar. You can see here, this is a constant rate IV. I think this was an hour infusion. So we can model that. You can see that the A's, the B's, and the C's that are in the 3-compartment model are really functions of a lot of the rate constants associated with movement between the compartments. And so, it's a little more complex. And I thought I didn't put it in the early one. What the program really solves for you is just the A, B, and C, but you can get all these other parameters, which are shown here, these transfer parameters between compartments, which kind of change the shape and why you get distribution and equilibration as things move in and out of the blood. That's really what you're measuring. The point here is that this compartmental model allowed us to start thinking about half-lives, how we predict them, how we predict drug levels, and we could start thinking about other dosing scenarios. And so, again, compartmental structure allows you to do a little more simulation and things associated with asking the questions about the pharmacology of the drug that you're working with. Here's another example with PI88, which is a heparinase inhibitor. And again, this was a phase 1 trial that was done here when Michelle Bashy and I think Scott Holden, Cindy O'Brien, Leah is actually an author on this one, and Samir Witta, a kind of who's who of the early days of the phase 1 clinic. And what this was a drug that was given subcutaneously. And so we had to model the absorption as a first order absorption, like an oral dose, like I showed you with the ABT. It also fit a 1-compartment model quite nicely. And you can see all the dose levels here from 80 to 315. We can model them all. This paper actually looked and started to do some kind of correlations between patient parameters and PK. We're actually able to show that this should be dosed mg per kg because it correlated with body weight. There's a lot of other kind of cool things. This data actually, along with some other data in healthy volunteers, was used to do some pop PK modeling, and I'll probably use that as an example for population PK modeling. We talk about that later to identify some specific covariates. We did that in this early paper here too, but then we did it in a more kind of a discrete and fashionable way using non-mem approaches later with some extra added data sets. But again, point is compartmental modeling to describe the pharmacokinetics of this heparinase inhibitor in early phase 1 development when we were doing it. Okay. What about some other um, modeling exercises? Here's actually, I pulled out a pediatric application, and this is actually looking at the interaction of cyclosporin A with etoposide and mitoxantrone in pediatric patients being treated for AML. Um, and there's a couple things. They did a control arm and a cyclosporin A arm. And the question here, what PK model was used for etoposide? Well, we have a half-life alpha and a half-life beta. So there's two half-lives, one for the distribution phase, one for the elimination phase. So this was done using a two-compartment model. And so again, if you have a little understanding of how this modeling was done, you can look at this and go, oh, okay, I understand what alpha and beta means. Alpha is the distribution, beta is the elimination. You can see that the average half-life in patients in the control arm was about 3.5 hours. When you put cyclosporin on board, all of a sudden it's 7 hours. So you get a significant, very significant, almost 100% difference in the half-life in the patients also receiving cyclosporin. So that could be a problem, right? If these are MTD dose, now all of a sudden you're seeing much longer exposures. You may be getting more toxicity, especially if it's involved with some type of minimal concentration that's involved with maybe renal toxicity or something else with these agents. And so it's pretty cool when you know that, this is the elimination half-life. Same thing here with mitoxantrone. Now you have alpha, beta, and gamma. If you were PK naive and hadn't sat through and been bored by this, you'd go, man, these betas are, this one it's changed, this one it's different. These aren't the same. But if you were a PK Rico Suave after seeing this lecture, you'd go, no, no, no. This beta elimination half-life is the same as this gamma elimination half-life in the three-compartment model, because that's how this was modeled in the three-compartment model with alpha, beta, and gamma. You could say, yes, with mitoxantrone as well, you're seeing significant differences in the elimination half-life and the elimination once the drug has reached equilibration. So more with a metabolism, which is what you would expect with cyclosporin A, because it does block CYP3A, it can block ABCB1, so it's blocking some of these transporters. And so again, having a good understanding of what these mean, maybe not that you can model it, but when you read the data, it makes more sense. Then you can really get a better feel for what they're trying to illustrate in this study, which is that cyclosporin A, when used with either of these drugs, changes the elimination half-life, which is shown here in this terminal phase, where it's much longer here with etoposide and also here with mitoxantrone. Okay, so being a PK modeling um, suave, knowing a lot about it can really help you, or being exposed to it can help you. Okay. What about some more kind of recent stuff? Here's some PK of pembrolizumab, so an anti-PD-1, or something that works the PD-1, PD-L1 axis here, an antibody. And I brought this up since there's a lot of biologic antibody therapies. And now, if you look, here's some data that was looked at from a number of actually Pembro trials. And this is a pharmacometrics paper. We'll probably talk about it a little bit later when we start talking about some population modeling again, because this was a population study. But they tested both 1 and 2 compartment models for Pembro using 2 mg per kg every 3 weeks versus 10 mg per kg every 3 weeks or 10 mg per kg every 2 weeks. So you can see if it goes out to 2 weeks here in these studies versus 3 weeks. And you can see that if you give it every 3 weeks, it doesn't go as high, which is what you would expect at steady state. Every 2 weeks, it does go higher as you model that out, because you're giving it, every 2 instead of every 3, versus what you would see with 2 mg per kg. The data was best described by a 2-compartment model, and that gets really important because they were looking at covariates in these Pembro patients. So, patients with different body surface areas, patients with different, fat content, blah, 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 whatever you're going to look at, size, weight, eye color, sex, whatever, you can throw that in there, and then you can kind of start making these adjustments to dosing that these covariates drive. You need to have a compartmental model structure to do that. And so in this case, it's a 2-compartment model, and they went through and did that later. So the compartmental structure is critical to the later analysis that they wanted to do. And so I thought I would throw this up to show that even with biologics like antibodies, you have the same type of approaches. These types of compartmental models can describe the pharmacokinetics of Pimbro, and they probably do the same with anti-CTLA-4 and with cetuximab and all the other therapeutic antibodies that are out there. So important to look at. Okay, I went over a lot of stuff actually in a relatively short period of time. I tried not to get too much into the weeds with the math, but I think it's important to note, most importantly, that compartmental models have structure. It allows you to kind of do some simulations easier. You can make some changes to the simulations that may be associated with various interactions. Like in this example, we're looking at metabolism, which is really manifest in the elimination half-life. So we can make some changes in that, see how much of a change you would need to get a significant change in the half-life of your drug or interest, and maybe even how much of a significant change you would need to make it become toxic if you knew what those levels were, to kind of dial in some of these dosing things without ultra complex modeling or actually doing those studies. So they allow you to make better guesses and to come up with better study designs than just going, I don't know. I mean, we can start guessing and say, if I did this much, we're going to be in a very dangerous spot. If I do it by this much, we might be pretty safe. So you can, come up with ways to do better study designs and come up with some kind of ways to simulate or think about drug interactions or drug combinations if you know how they interact, or you think you know how they interact, or they can't interact with this level, you're going to have a lot of trouble. Okay. So I'm going to wrap this up by talking about non-compartmental versus compartmental. Why use one versus the other? Well, for descriptive data, it doesn't really matter because you can calculate AUCs, you can calculate half-lives, you can calculate blah, blah using either. Okay. So for descriptive data, either non-compartmental or compartmental can work. All right. If you want to simulate altered conditions, maybe simulate the effects of pleural effusion altering the volume of distribution. So maybe you have a therapeutic antibody that's really distributed in total body water, but you have somebody with a large pleural effusion. Now you have this increased liquid, and they're on these, you're um, pumping them full of fluids because of this pleural effusion. You know, you might be able to play around with the volume distribution. Go, wait a minute, if I increase this by 30%, what's going to happen to my therapeutic antibody levels? Or now we're going to be out of the range. So you might be able to, figure out ways to calculate a dose for people with a little bit altered drug distribution or drug metabolism. And so you can play around with that. And we've certainly done this. I have a great case study with a dog that had a post-surgical infection for head and neck tumor that we use in veterinary pharmacology, and the pleural effusion changes. I think it's Enro. I think we're talking about enrofloxacin or something like that. It changes its distribution. No, it's an aminoglycoside. Changes its distribution such that you have to give higher doses because the drug sits in this kind of pleural space. And so it is something you can kind of play around with, and you can get more effective dosing if you kind of alter these conditions. And compartmental modeling allows you to do that because the volume distribution and things are inherent to the simulations. Um, Simulate different dosing scenarios, sub-Q or longer versus shorter infusions, or maybe you have a different formulation that's going to change how long. It's going to change your Ka. You can start doing that as well. So you have these values for absorption that you can start changing as well, and you can actually, am I going to get lower or higher? If you're doing, think about when they first started using doxorubicin, and they realized if you give it as a bolus IV dose, you get more acute cardiac events. So they started giving us as a shorter infusion, or relatively short infusion, 5 to 15 minutes, because it distributes so fast. You could actually simulate that. You can give 30 mgs per meter squared, give it over 1 hour versus giving it over 20 minutes versus give it over. You can simulate that pretty easily and see how high your max is. So if you have to avoid a certain max because you may see some toxicity, you can play around with these different dosing scenarios, and the compartmental structure gives you the structure to do that and the parameters that you would need to do that. So you can fiddle around and work out, what if I lengthen the infusion time to an hour? What if I shorten it to 30 minutes? Are we going to be safe with concentrations, or is it going to be a problem? And we can test covariates, and this gets more to pop PK modeling. This is going to be a whole separate lecture or series of lectures that we can talk about. bringing in population characteristics. And to do that, you have to have a compartmental approach. You have to have structure. Okay. All right. What I hoped I convinced you of is that compartmental modeling is not just curve fitting exercises, but there's a lot that we can learn from the pharmacology and the medicine associated with these that allows us to kind of start to do some simulations. So with that, pretty small group, but I can gladly take any questions. And I know Jill's going to throw this up for people to look at. Anybody that looks at the recorded version, if you have any questions, feel free to email me and we can talk about this at any time. So, all right. Any questions? All right. With that, everybody get ready for Snowmageddon.